prepare for the cybersecurity point of view. And sorry for that, but I
No,
just
no, no
prefer the
problem. Means
machine
our
learning
student
and
also
AI.
can know that cyber that agentic
Right.
is not only for the this one only cyber security. We can
Only for the
apply it.
cybersecurity. It can be also in terms of computational biology or bioinformatics like that. So let me share my PPT. Uh,
Yes,
then
yes.
I will
I have
start
already
it.
made you the co-host. You can start.
Yes, sir. Is it visible now?
Yes, yes. It's coming. Your screen has already shared, but PPT is not there.
PPT is there?
No, no, no.
No. Is it visible?
Screen is visible. I think you have to share the whole screen or PPT you have to share.
I have shared the whole screen.
Open the PPT then. I
Present
think.
something else in their screen. Okay. Is it visible now?
Your screen, yes, yes. Now it's coming.
Mm.
So cross that one. Cross that small window and make it full screen.
Mhm. Now it is full screen. Okay.
Cross this one. You can cross it. Huh? Yes.
Now it is
Yeah.
visible, right, sir?
Yes, yes. Yeah.
Mm.
It's-
Thank you, sir. Thank you for the introduction as well as the topic, whatever I am going to present here. Actually, related to the machine intelligence that used to transfer the different uh, techniques or technology what are used previously. Now, in terms of AI or machine learning or deep learning, and now the question is related to agentic AI or explainable AI or generative AI. So these are the new terms which are associated with all the things means artificial intelligence. So all the problems which are handled by using different technology or techniques without intervention of human being also, either it is mechanics or physics or whatever it may be. But now everything is converted towards to the AI, artificial intelligence. So my point is here simply methylation site prediction. So as we know that the computational biology related towards to the problem of human Homo sapiens, or you can say any creatures throughout the world. Here we are trying to I am trying to consider related to the Homo sapiens or human being. So a human being itself is a machine, we can say, and to understand the behavior or we can say the functionality or technique, whatever used in our body. So one of the thing is what methylation site prediction. So methylation itself is a process which is takes place in your body, either it prevention towards to the disease or to detect the different malfunction which are going to be held in your body in root level, or you can say intrinsic level, that is either in DNA or protein level. So these are the things which are manually done previously. So now what happened, and due to the huge means the problem is a exponential problem. That this problem is also exponential problem to check whether this particular portion of the DNA sequence or protein sequence is methylated or not. Methylation is a process, and a particular portion of that particular sequence is methylated or not. That need to be predict, and that prediction initially takes place by using laboratory, and which is a very expensive procedure to do all the things. So now how machine or you can say artificial intelligence or explainable AI or agentic AI can help us to do but to predict or to identify whether this particular methylation is occurred or not. If occurred, then whether it is positive or negative. If positive, what happened? Then if negative, what happened? So nowadays, simultaneously, each and everything is going to be automated. That means initially we take the blood sample, then we have to convert it into the sequence, then we have to check it like that. Now each and everything is going towards to the automation point of view. So one of the technique is agentic AI or explainable AI related to this particular methylation site prediction. Anyway, I'll come one by one related to that, whatever the different models used for this thing, and what is methylation, how computer science or we can say AI, machine learning need to be used here, And what is this problem? Why it is a computational problem? If it is a computational problem, only we can think about the machine learning or AI technique need to be used it or not. So anyway, this is my profile related towards my collaboration as well as working at NIT Srinagar, NIT Jalandhar, as sir already introduced. So we have some collaboration in within India and outside India also related to this topic and other topics. And these are my PhD students which have already awarded. 4 5 students already awarded. Uh, this is the thing which need to be understood, because intelligence, that means if you say that natural intelligence as well as artificial intelligence, so these are the different uh, um, different enhancement of the intelligence. That means we are not saying that we are now using the intelligence, but previously also the human intelligence was there from, and we are able to learn from the natural intelligence. So due to the evolution of the human beings from society 1.0 to society 5.0, similarly from industry 1.0 to 5.0. So these are the different involvement of the machines in different situations. So based upon that, so we are at industry 5.0, and also we can say we are at society 5.0. Anyway, here the two things related to this natural intelligence as well as artificial intelligence. So natural intelligence was there. That's why artificial intelligence here. So natural intelligence, it related to the human brain, then learning from the experience, emotion, and creative and flexible of thinking. And artificial intelligence means the computer system or learning from the data, and the different algorithms are there, models are there. Then we are trying to forecasting the uh, process, or we can say past the processing, existing processing. So after this artificial intelligence, then if you go for the artificial by agentic AI, by agentic artificial intelligence. So here agentic AI refers to AI system that can act autonomously towards the goal rather than only responding to a single prompt. So this system behave like intelligent agent that can understand the objective. That means if I just want to consider this problem, methylation site prediction in terms of from the computational biology point of view. So that means my objective is to predict the methylation site, whether it is positive or negative. This is one objective. But after identifying this objective, whether it is predictive, positive or negative, then what is the implication of this positive? If it is positive, then what to do? What happened? If it is negative, then what to do? What happened? That means if that particular whole system can identify it, can generate the sample, can identify it, whether it is positive or negative. If it is positive, then what to do? If it is negative, then what to do? Then the whole system is said to be agentic AI. Then we have to plan the task, what to do, then make the decision according to the objective. Then we have to use some tools, whatever the tools are available. Then we have to interact with the environment based upon the other features which are to be associated with that particular point of view. Then we have to also learn from the feedback. If you do something, then we have to learn from the previous experience and feedback whatever given by the agents also. So then we have to execute the multiple steps workflow with the minimal human intervention towards to the objective of the problem. So unlike the traditional AI chatbots that mainly generate the response, so agentic AI can be independently perform the different actions and coordinate complex processes like computational biology. I'll go to directly that particular point. Here we can say, if you want to see that to build a agentic AI machine, so which behave like a human being. So as we know that there are different response for the human being. One is skin, tongue, nose, ears, and eyes. So all the things are takes the input, and these are the different sense organs from the human being which are used to take the input, like skin. When you touch something, then pressure, temperature, it can be related towards skin. Then the tongue, we can taste something by using nose. We can smell something by ears. We can sound some, receive some sounds, and from eyes we can visualize something, or the whether the related to that object. So all the input signals, whatever the different organs are there, if all of them are related towards to take the inputs from the environment and process it and finally we will respond it. So like agentic AI, we are also trying to do the same things. So if one organ can be implemented for a particular point of view, so it can behave like that. So here directly I will come to the point, the protein methylation. Uh, the proteins methylation site prediction in terms of computational biology point of view. Here, the proteins are made up of hundreds and thousands of smaller units, we called as amino acids. So, which are attached one another in a long chain, like this figure. The first figure related to different characters are there. The amino acid sequence, there is the primary sex structure. Then we have to go for the secondary structure, then tertiary and quaternary structure. The quaternary structure, due to the tertiary structure, our body, but the nature in the nature, that 3D structure is there because of this tertiary structure of the protein sequences. Here, the proteins are involved in the most of the cell functions. There are different functions which are expressed in your cells, so such as metabolism, hormonal regulations, transportations, and the control of the genes. So there are different functions which are involved with the protein structure. So here what happened, after the protein synthesis, additional modification may be required on the required to the obtain the structural and functional variation of the proteins. So these modifications are a post-translational modification, we can say PTM. So this is a process which takes place. So in protein methylation, proteins are enzymatically changed by adding methyl group. That means there is a group of protein that is methyl group. So here we have taken two groups, that is arginine and lysine. So arginine and lysine are the two amino acids which are present in the protein sequence, and the methylation process takes place within that protein sequence, which is used to involve for the DNA repair, generative activation, and gene slicing. So these are the different functions which are takes place, and based upon these two amino acids, that is arginine and lysine, so the methylation takes place. Here, the arginine methylation happens when the R or G is region impacting the protein-protein interaction and protein structures. Whenever the position, in any position, as per in the previous structure, at any position, if the R, the character R, that particular amino acid is there, so though we can say that it is R methylated, particular R methylated group for that particular protein sequence. So methylation and their regulative enzymes, which are likely to be a wide range of human diseases. So basically, different diseases are depends upon this methylation site whenever it takes place. So it related to the disorders or such as neurodegenerative disorders, coronary heart disease, cancer, then arthritis, then multiple sclerosis, then due to their role in the different gene regulations. So identifying this methylation site is required to compare the molecular structure of methylation site is better than in terms of mathematical point of view, in terms of wet lab point of view. So for this figure, if you see that, okay, this is the DNA sequence which are consist of the amino acids. Then from the sequence, the DNA amino acids there are 20 different amino acids. Out of them, all the characters 20 characters are used to represent those 20 amino acids. So M, K are like that. So our target here is R methylation. So we have to identify from this protein sequence, we have to identify it is a large sequence in terms of computer science point of view. In terms of, let it be, the length is 1000 characters are there. So 1000 length of a protein sequence is 1000, let it be. Then this 1000 length, the characters are from the 20 characters. A repetition of those characters, it makes it 1000 length of the particular string. So within that string, we have to identify how many number of times the R is there. So how many number of times the R is there, so that much of number of time R methylation takes place in that particular sequence. So out of those R methylation, there are two types of, maybe it is positive, one of the position may be positive, maybe negative. So those things need to be identified, whether it is a positive methylated or negative methylated, like that. So these are the arginine methylation group. It is related towards carbon dioxide, then NH3, CH3, like that. So these are the 3D structure we can say of the arginine methylation sites. So based upon that, there are basically positive or negative. So it depends upon the positive or negative. If it is negative, it towards related towards the different disease like cancer, Alzheimer's disease, then other diseases also responsible for this particular point of view. Here we have to identify the R. Here the whole procedure related to the identification of the R, where R is present, whether it is positive or negative. So there are different samples we have taken for the methylation point of view, and here also we have used 4 different species like human, chimpanzee, mouse, and rat. So all of them are involved with this methylation process. And our identification is what? For human, the data set is given, then for chimpanzee, mouse, and rat. So there are some negative or positive samples, which is asymmetric dimethylation, then dimethylated arginine, omega methylation arginine, and symmetric dimethylation. So 4 different types of methylated groups of 4 different species have been studied in this particular study. Now the question is why this thing can be considered as a computational problem. Here the experimental methods such as yeast to hybrid, there are basically 3-4 methods which are wet lab point of view. So one of the method is hybrid system, then mass spectrometry, then protein chips, which are lab and labor intensive and time consuming and expensive. Because if you know that if 20 characters or 20 amino acids are there, if you do it, if the length of the sequence is 1000 or 100 only, so to make the combination of this 20 for 1000 times, it is very difficult to find out all the possible cases. So a protein sequence, for example, if a protein sequence is made up of combination of 20 amino acids only, given, then if a protein sequence length is n, like my question is, if it is 100, then if the length is n and 20 characters, computer science point of view, if you're trying to understand, let it be there are 20 characters, and we want to generate a string from those 20 characters. The length should be 100. So that means what? Now 20 to the power 100 number of sequence can be generated. So 20 to the power 100 number of sequence which are to be generated, it is not possible to generate in supercomputer also nowadays. So how to perform all the combinations in wet lab, means lab test point of view. So it is very difficult, or we can't do all the things in case of lab. So then what to do? Then we have to treat it as a computational problem, and maybe the AI and machine learning techniques to help to find out this problem related to that, whether it is positive or negative. So as I told that if a protein sequence of length is 100, here if it is n, then there are 20 to the power n number of sequences which are possible for a computer to process all the sequence. So it will not take a long time. It may be days, months, or years. So the machine learning helps in processing a sequence of finding similarity and other features that distinguish them from the other sequences. Here different data sets, like the data set from the protein methylation is provided by the author Deng in 2017, that is also given. Then the data set consists of 3,900 arginine sites here and 1,751 proteins for the positive, and 86,111 negative sites for the arginine methylation. So that data set is already given from Uniport or PDV, protein data bank. These data sets can be collected. So these data sets, whichever is given, and also we have created some data sets from PDV, protein data bank, as well as the Uniport data bank. Here, if you see that this particular sequence EEMTSR DYYFD. So this is a sequence, but we can say this is a subsequence of a particular protein sequence having length 11. Here the R is present in the middle, and before R there are 5 characters. After R also there are 5 characters. So it was observed that to identify the methylated, if it is methylated or not methylated, based upon that. So it was observed that if you take the previous 5 character and after 5 characters, then based upon some physicochemical property or mathematical statistical property, we can identify whether this particular sequence is positive or negative. Similarly, for this sequence, this is R methylation and K methylation. Basically, there are two types of methylation, R methylation and K methylation. So in R methylation also, we have considered 11 length as the substring of the from the protein sequence. In case of K methylation is also, we have taken 11 length of the strings original protein sequence, whether it is K methylated or R methylated, both the cases, the length of the subsequence is 11. This is another 3-4 data sets which have already there in some of the papers or literature we have taken. And also we have created some of the data sets from this website, that is Uniport KB. See, before implementation of any type of AI algorithm or machine learning algorithms, we need to have some data sets which need to be standard data sets. So and those are the data sets which are standard data set used by different researchers in literatures. And simultaneously, we have created some data sets from this Uniport website, and that is which is considered the standardized data set related to this methylation process throughout the world. So this is the Uniport KB data bank from where we have created the data sets for Homo sapiens and mouse, then rat and chimpanzee. So all the data sets are created from this particular web pages. So this is the example of the data set. Then the first one is the ID, protein ID, then the protein sequence. You can able to see that, okay, there are different characters, M, A, T, D, like that. So in the sequence, these are the data which are generated from the lab and kept in the web pages. And after that, they have identified, okay, some of the portions which are there as R or K are the methylated. For this particular sequence, the protein ID Q9WVG6. So the sequence is given, the length of the sequence is 551, 551, and R is there, and it is dimethylated arginine, and which is the sequence of Homo sapiens. So it is a dimethylated arginine as R present various different positions. The length of the sequence is 551, and it is a dimethylated arginine, and it is belongs to the Homo sapiens. That means for the human being. Similarly, all the sequence, some of them is asymmetric dimethylated arginine, and some may be symmetric. Some of them may be omega and methylation. So like four different types of methylation with 4 different species have been given in this particular data sets, and which are generated from the data sets based upon the information available in the web page or website. Then once these data sets are created, then we have used different statistical methods to extract the features and to learn the patterns, or you can say the different rules and related to that particular sequence. So one of the features which are extracted by using the Shannon entropy, which is a very known mathematical parameter to extract the information from a signal from a particular sequence. So Shannon entropy, as we know, that it is used to determine the how much uncertainty or information a sequence can contain. So here, if the sequence is 1,100 or 2,000, whatever it may be, so that particular sequence have some information based upon the number of characters present in that particular sequence. If you say that a protein sequence is consist of, suppose, 9 times A, all A. So if it is all A's, but all R are there, arginine is there, all R is there, then what happened? That means there is not much more information related to that, because everything is R. So that is not important towards to find out that methylation site. That means the information which contained by the sequence which contain all R, whatever the length, it may be 10, it may be 11, it may be 100. So there is no such information. So the question is, if that 20, if the length of the sequence is 20, and if 20 characters present, that it contains more information that each and every character is present in that particular sequence instead of one character. So that is considered as one of the features which need to be extracted. That what is the uncertainty or information content by that particular sequence. So that extracted by using the Shannon entropy. And here we have considered the Shannon entropy, and 20 characters are there, I is equal to 1 to 20, and pi means the probability of a character, and log base to p of I means the probability of that character which is given. That means here 20 number of characters are involved for this particular problem. So this 20, each and every character, that is the repetition of this character, then we have to find out how much number of times a particular character is present, and uh, the 20 characters information need to be calculated for each and every sequence. And finally, by using this formula, we can able to find out the Shannon entropy value of that particular sequence. So if there are 100, in that case, the 3,500 number of sequences are there. So for each and every sequence, the Shannon entropy is to be calculated. Here, the pi indicates the probability of the ith amino acid in the given protein sequence, and we have to find out it. Next, another point is the first exponent. The first exponent is often used to analyze the autocorrelation that between a time series data analysis. So here also we have used the host exponent related to these parameters to extract the information from that particular sequence. Same thing related to this fractal dimension point of view. This one is also one of the mathematical parameter, and here the mathematical parameter which are used for this for this particular feature. So here the each and every character, so it can be converted into a 20 cross 20 the length, as per the length of the protein sequence is given. For this case, 11 is the length, is considered the 11 length. In the middle, the R or K need to be there, and before R we have to take 5, and after R also we have to take 5. So that means here a matrix can be generated which is 11 cross 11, and what are the number of characters are there. If the characters is same, we have to put 1. If the characters are different, we have to put 0. Then a matrix will be generated. So by using this matrix, then it converted to image. Through that image, we can able to find out the dimension, fractal dimension of that particular image by using the box counting method, which is again a mathematical parameter for that particular sequence. Then along with these 2 or 3 statistical parameters, so there are some other physicochemical properties of the amino acids which are used in different web server also, and for us also. Here the physicochemical properties such as the extinction coefficient, molecular weight, aliphatic index, instability index are calculated in this particular research point of view. The extinction coefficient, which is used to demonstrate how much light a protein consumes at a specific wavelength. So there are some tools and there are some formulas which have been implemented to calculate the different properties. Similarly, for instability index is also calculated based upon the protein sequence. So for a protein was calculated using the following equations, and where n is the length of the protein sequence, here n is equal to 11. Then the prediction of the protein methylation sites based upon the various statistical mathematical features are extracted from the protein sequence. So, and multifactorial feature extraction site and prognosis model is also for the prediction of the protein methylation site is here used. So the proposed model, which we are exploits the sequence-based features, are all of the sequence-based features, physicochemical properties, that is amino acid composition, dipeptide composition, and conjoint triad features and entropy. So these are the 5 different features are used to convert those particular sequences into in a mathematical or feature point of view. And after that, we have used different machine learning algorithms like decision tree, random forest, Navier, then deep neural network, and SBM. So this study also explored the impact of the arginine methylation as well as the lysine methylation. Both of the methylation we have takes place. At the end, I will tell you actually these are the process. These are the feature extraction or phase feature generation from those protein sequences. Then these are the algorithms then these are the algorithms and the feature extraction. All of them are implemented in a web page where we can input the sequence, and that particular web page we can give us the information related to that, whether it is methylated, or if it is methylated, whether it is positive or negative, like that. So then the proposed architecture will look like this. We have first collected the data, then the feature is representation related to the amino acid compositions, then entropies, dipeptide composition, then CTF related to that particular feature extraction. Using these feature techniques, then we have converted a particular sequence into 777 number of features. That means a particular, if there are for human being, the positive and negative, if you see that, okay, 1800 positive and 500 something data are negative. That means 2300, 2300 number of sequence for the human being was there. So, and for each and every sequence is converted to a point of features, that is 777. So that means it can be treated as a matrix which is related to with the dimension that 2300 cross 770. So those 2300 number of protein methylated sequence is converted into a feature matrix with the dimension 2300 cross 7770. And each and every features related to these 5 different types of meta features: amino acid composition, protein peptides, entropies, dipeptide composition, and CTF, for information theory point of view, that particular sequences. So by converting those sequences into different feature representation, we have represented those features. So similarly for all other species like chimpanzee or mouse and rat, then the training data is there. Then there are different algorithms like random forest algorithms. Then we have implemented it, then testing it. So based upon this architecture of these models, then we have seen that, okay, the performance related to decision tree, SBM, Naviverse, or DNN, deep neural network, random forest. So random forest algorithms, it generate better accuracy related to these particular data sets than other. And this is related to the previous one is arginine methylation, and the next one is lysine methylation. So here our random forest is also working better than that. And these are the RFC curve for different classifiers which are used for this particular arginine lysine methylation sites. Then the impact of these features, all the features which are generated here related to the amino acid composition and protein peptides. So all the features are there, but all the features are not equivalently by equally contribute to the classification. So here, what happened? Now we are able to see that, okay, what are the impact of each and every feature individual based upon those data sets. So then individual features are taken as inject to the machine, but to the model. So we are able to see that amino acid composition gives this much of 75.28% of accuracy, whereas the other features, protein peptides, then enzymes, DPC, dipeptide composition, CTF, these are the features which are given the different contribution towards to the find out the accuracy of the particular system. Whereas if you include all the features, then the accuracy is better than individual features. Even if the amino acid composition and protein peptides, EN, then DPC, whatever the features are there, when we include by all the features, when we add all the features and taken as input by the machine learning algorithms like a random forest algorithm, then it will give better result as comparison to individual features. This is similar for the lysine methylation datasets. And after all, whenever we are comparing the proposed model with other datasets, dataset 1 with other models. So also the proposed model related to the arginine lysines, the accuracy is given as comparison the previous model GPS MSP of one of the paper in the literature also. These are the another comparison related to the different datasets. Two related to the methyl PAC and MEP PRED and RF algorithms, which are given by other researcher. So for our model, it gives better than that. Those also somehow some point it gives better for arginine, and for lysine some other models are better than them. So this is the result related to the different literatures which are exist and proposed different models like MEMO, MASA, BPV. So the references are there. So these are the different models. 7-8 models are there which are already exist for this particular type of problem. And our model is also implemented upon those data sets as well as those algorithms. So the accuracy is related to this 93 point something. After this is the general machine learning techniques which have been used for this particular problem. But here what happened, after that our target was related towards the use of explainable AI in case of all the sequences which are there, and how can used by how other researcher can be used, so that we have developed a web page website for this particular problem also. Here to interpret the predictive machines of the prediction model using explainable AI, we have used. And this the proposed model, PRMXAI, exploit the sequence-based features, whatever the features we have already discussed. So including the protein peptides, DPC, then AAC, amino acid compositions, and some information theory-based features, ITB. So all of them sequence-based features are used, along with the explainable AI is also used. Here, the proposed model interpretation is also showing the use of explainable AI, artificial intelligence. And the method was used, SAP method is used for this thing. Safely additive explanation method is used for the AI point of view, explainable AI point of view. Then for this thing, the proposed model is look like this. That means you have extracted data from the uniport. You have considered two types of methylated. One is monomethylargine, another is dimethylargine. So in case of monomethylargine, there are positive sequence and negative sequence. So 3,551 are positive and 84,000 something sequence are negative. And out of them, we have considered 1,465 sequence for positive and 39,000 sequence as negative. Then it is converted to as a original sequence from the original sequence after pre-processing. Then we have converted. Then based upon those sequence, we have used the features which are amino acid composition, dipeptide composition, and physicochemical properties. Then information theory based features. So all of the features, that means those sequences are converted by using these features for feature vectors. Then based upon those feature vectors, the models like SVM, decision tree, random forest, k-nearest neighbor, Navy-Bayes algorithms have been used. Then out of these, by using the 10-fold cross validation, we have checked the accuracy. Again, we have upon those models, we have also implemented that case of XI, explainable AI. And the accuracy related to this is 87.17, and rest of the parameters is also as shown here. I'll come to the point. Here, the model interpretation using explainable AI, what happened? Now, whatever the number of properties, like feature extraction properties are there, or feature generated. So all the features are to be treated, as well as whenever the XAI SAP method is used. So we are able to see that, okay, the amino acid composition here takes the major role, that means towards to the positive and negative. That the role of the AZ, amino acid composition, and first it takes towards the classification towards positive, negative towards negative, then DPC, and molecular weight is also there. Amino acid composition E, then DPC, all the parameters are there as explained here. So each and every parameters or features have a contribution, but the major contribution is the amino acid composition part in terms of any and information theoretic like Shannon entropy or Harvard entropy, Rennie entropy, whatever the number of entropy related uh, parameters have been implemented, features implemented. So all of them are there, but the amino acid composition takes major role for classification towards to the positive, towards to the negative. Then this is for the model interpretation related to the feature importance for the prediction of the dimethylation sites. Here the summary, this is the plot related to the SAP value and the features average impact on the model prediction is there. Anyway, to do by doing all the things, finally we are reaching at a point that the protein arginine methylation by using the explainable AI proposed method, that is monomethylated and that dimethylated, that is for monomethylation it is 87.17 and for dimethylation 90.40, the accuracy is there. And these are the already existing model. Again, we have compared by using this explainable AI case. Then this model is this related to this. Here the deep vision model, whatever the model of algorithms have been implemented. So all the algorithms have been implemented as a web page like that. So here the prediction of protein methylation sites using the deep learning algorithms with the help of various encoding schemes. Here the experiment to find the window size of this peptide sequence length is given, and the web page is like deep pprms nit.ac.in. So from here only, I can come to the point. From here only, we can able to predict this by using this particular web page. We can when we give a input a particular sequence, it can able to detect how many number of positions are there for positive and how many number of the positions of the R are negative. Then how many of them are positive methylated and negative methylated, whether it is methylated or not first, then whether it is positive or negative. So through using this web page also we can able to interact. So this is the complete architecture of the deep protein R methylation site prediction. And again, this is converted into the amino acids again converted instead of treat each and every amino acid is one amino acid. Here, what happened based upon some of the side chain and dipolar strength of a particular amino acid. So we have make them group. That means the 20 amino acids again converted into 7 groups. So A, B, G based upon the property of the side chain as well as the dipolar strength are belongs to one group. Then APLI are the protein are the amino acids which are belongs to another group, that is second group. SMYT is one of the group. QNWH is one of the group. As computation point of view, we have treat each and every amino acid as a character, and each and every character is represents amino acids. And based upon similar property, now first it converted to into 7 classes. So based upon the property that C is in one class, but R is related to the K because our target is R methylation, K methylation, both have the same property. Based upon this only, side chain as dipolar strength. So both of them belong to one class, that is the 5th class. Then by using the architecture, CNN architecture, we have trying to, by using deep learning CNN architecture, we are trying to find out that methylation site problem. It is a 2-class problem, methylation site, non-methylated site, or we can say positive or negative. So again, the GRE model architecture of the prediction of the methylated site based upon the architecture. So also we are able to predict. So this is the proposed architecture to identify the protein methylation sites. So these architectures, theoretical point of view, if you all of you know something, or I can explain this thing also based upon that CNN architectures, how it can be converted from the protein sequence to a full connected layer of ANN, artificial neural network, CNN, convolutional neural network. So this the describe the hyperparameters which are used in the model, and based upon these architectures, we are able to find out the accuracy as mentioned in the particular table here. Then again, as initially I told you that we have considered there are different window size. The 11 is the standardized size. So throughout the literature, here we have considered the window size as 11, 15, 19, 23, 27, 31 up to 55. Here we are able to see that the window size is 51 because the R is in between, and 25 in the previous to the R and 25 after the R. So the length is 51. So the length 51 tells us gives us better accuracy for this deep learning concept instead of machine learning. That is 83.38. So we reported this all the results in the research paper also. So this is the performance result related to accuracy, specificity, and sensitivity of all the models. Here the encoding scheme for this particular architecture is used one at encoding, indicator matrix encoding, and group-based coding. Then embedding the encoding, the 4 different types of encoding scheme are used. That means the sequences are converted to a image point of view based upon these 4 encoding method. So out of these methods, so embedding encoding generate the better result as compared to the other. And we give the name of this proposed method as deep PRMS, and this the accuracy for this particular model is 85.32. And to this is the web page. This is the web page related to that. That means this is the tool we can say which is there. Simply by using this tool, what to do? We have but somebody can give the sequence in terms of in the FASTA format. When we give it, then we call the feature extraction method in the backend, and as well as the algorithm in the backend. Then it will gives us the information whether this is methylated or not. If methylated, then whether it is positive or negative. If positive, then what should be the treatment, or you can say course of action need to be taken. And if it is negative, then what is the impact of this particular methylated site or position, and what should be the next course of action need to be taken by using this website. So here for the example, the sequence is given as input, and this is the output. The output is related to that. Okay, this is the protein sequence, that is only one sequence, and there are different positions where the methylation site takes place, and the score is also given. The score is like 83.83, 80%, 89, 90. So these are the score for the 40. Only one sequence is there. One sequence sequence is given as input, and where is the methylation takes place, that positions is also given, R positions. And for if it is given R position is identified, then how much score related towards to the prediction, that also given prediction score. But after this, the agentic, how the agentic AI can be work here. So actually our expertise is related towards the computer science point of view or computational point of view. So if some of the doctor, or can say researcher related towards this point of view, those who are working lab will involve. Actually, 3-4 months I had a interaction with one of the professor from Central University, Bhatinda, Punjab. So with him also we are trying to validate this process in terms of lab point of view. That means if some of the sequence can be generated by them, Central University Bhatinda as well as AIMS Bhatinda. So, and me also, with 4 groups are there which are working in wet lab point of view. So whatever the algorithms or feature extraction techniques we have used and we have implemented this point of view. But after this, what to do? After this, what to do? How we can automate this thing? That means if the sample will come collected, then this need to be converted to a sequence, a protein sequence. Then the protein sequence need to be converted to based upon this algorithm. Then after that, it gives the score of the methylated, whether or methylated or not. After that, what is the next course of action need to be displayed by using this particular tool. So that is our target. So this is the research related towards, I think, 2024, 2023. Before that, After joining that NIT Jalandhar, I had a contact with AIMS Bhatinda as well as AIMS Central University Bhatinda. We are trying to do some wet lab research based upon the studies that are available, and also we are trying to include all the information to this web page. Hello?
Yes, yes.
Yes, sir.
Uh,
Hmm. And uh, here the role of agentic AI takes more major contribution than only machine learning or deep learning. So point is that if we will do the things from initial point to the samples to the decision, so we can say the role of agentic AI that this can be treated as a tool, or it will pre-process the data, generate the feature, implementing in the deep learning algorithm, and identifying the methylation, then identifying the score. Then if it is positive, what is the next course of action that need to be display here, and if negative, what is the next course of action need to be displayed here. So the full-fledged project was not yet developed. I can say, or not yet developed. The research is still going on. Our target is that how to do this thing, link this computational biology or this computational parameter to the lab, wet lab. So up to here, we are here. We can able to predict the score if the data set is there. But after that, what to do? So that is also the work is also going on. And here the conclusion is related towards the method based upon the sequence information only. Yes, this is one disadvantage for this method. We are targeting only the sequence point of view. But yes, we can include the structural point of view, 3D structure or pattern structure. Information need to be involved with this particular tool, which can act as agentic AI for this particular problem. So extract the informative and distinctive feature from the amino acid sequence, and a type-specific predictor was proposed, the prediction of the protein methylation site for this particular problem, and which is useful to understand the biological significance of the methylation sites. We can introduce the classification strategies based upon the deep learning algorithm to reduce the bias introduced by the feature extraction. That means all the features are not important, or maybe some other features can be defined to identify these things. Other features can be defined. Still, some of the nowadays here 2-3 MTech students is also working with me related to this by using transformer. That means instead of treating that, okay, this is sequence, then converted to a mathematical using mathematical parameter decoding or encoding process, we have converted to that sequence into a mathematical parameters. Then we have to use it to the machine learning or deep learning algorithm. Now the question is, by using the generative AI transformer, that means if you treat that, okay, that particular sequence is a language for this particular type of process. So if A, then TRI, then T, then GRI, like that. So by using the transformer, what are the current transformer used to understand the protein language? By understanding the protein language, how we can classify this thing instead of using any handmade features like that. So students are also working with me related to that transformer, or we can say generative AI. So this website is there, and up to this, yes, up to this only we did, and we are trying to make this tool as a agentic AI towards to this particular problem, that is methylated site prediction. Okay, thank you. If any doubt, please let me know.
Yes, open for the questions. If anyone having doubts, agentic AI you want to apply in medical domain or your computational biology or bioinformatics, then you can directly contact to sir. And sir is not working only in computational biology, as I told that he has also having other fields also, cyber physical system, then information security. So in that area also he is working. As in a very short notice, sir has not able to prepare the this for this presentation in computer cyber security, but you can contact to sir directly, or I'll
Yes,
talk
yes.
with you so that anyone is interested, then we can have collaboration with sir.
Yes, sir.
some students of the sir, those who are working. So if any questions, then open for the questions. Otherwise, we will stop. Thank you, sir.
Thank you, sir. Thank you.
I've already watched the whole session. Interesting one.
Hmm.
Any questions from anyone? Like Aparupa, whoever is Bhumika, Bhumi, Siddharth. Anyone having any queries, you can ask. All of you are there or not?
No, some of them are there.
Okay.
Okay.
Okay
Sir.
then, Nanjin sir, thank you. Huh? I'll contact
Thank you,
with
sir.
you.
Okay.
Thank you.
Thank you, sir.